So when Barry asked me for a title, I thought I have to incorporate Star Wars. And I changed the Star Wars icon to be SARS Wars. These are coronaviruses. Here, coronaviruses are called coronaviruses because they have a little crown of their spike proteins, which we will come back to today. So the coronaviruses are a rather large family that affect a lot of vertebrate species. There's 7 different types that infect humans, 6 species of them, and then there's 1 species where there's 2 family members that both infect humans. And one thing that's very interesting is that SARS-CoV-2 has greater nucleotide sequence identity to bat coronavirus than it does to the other human coronaviruses. So you can see here for the common cold coronavirus, 50%, for MERS, 55% identity, for SARS-CoV-1, only 80%. The bat coronavirus, it's 96%. And it turns out that China has a very diverse and rich fauna of bats, and also has many, many coronaviruses. There's at least 70 different species, or yes, 70 different coronaviruses that have been identified in bats in China. So today, what I was hoping I could accomplish is to give you an overview of the mammalian immune response to pathogens, talk about innate immune system detection of RNA viruses, how coronaviruses infect us, the routes of entry and dissemination. Because they enter at mucosal surfaces, I'm going to talk a little bit about mucosal immunity to pathogens. And I think, have all of you heard of cytokine storms?
I mentioned it a little bit in saying that that would be one of your topics, but we don't know much at all about it.
So I'm going to talk about cytokines and cytokine storms, what they are and why they happen. And then I'm going to talk about SARS-CoV-2 vaccine strategies. Originally, I'd been planning to talk about all of the different strategies, but since the Moderna vaccine and the Pfizer vaccine are both mRNA vaccines, I thought I'd focus on those. So first, let's talk about how the immune system first gets alerted to a pathogen. So a pathogen, antigen is a term that immunologists use just to talk about any molecular entity that can elicit an immune response. So by definition, bacteria and viruses are antigens. And this first gets engulfed by dendritic cells at the tissues. So for example, viruses that we inhale, influenza virus, SARS-CoV-2, MERS, SARS-CoV-1, these all enter through the airways, and we have immune system cells that are particularly dendritic cells that can take up the virus. This launches a T cell response. So the immune system consists of 2 types so let me backtrack. The immune system has 2 arms. One is the innate immune system. A very important member of the innate immune system is this dendritic cell. And then there's the so-called adaptive immune system, and that consists of both T cells, which we're going to talk about today, and B cells, which give rise to the cells that produce antibodies. So a dendritic cell brings in the antigen, stimulates a helper T cell. That cell, if it has a receptor that's specific for any molecular entity on the antigen, will proliferate and differentiate. And it'll produce cytokines, which I'll come back to later. These are molecules that stimulate proliferation and differentiation. So it'll produce an important cytokine called IL-2 that auto-stimulates it to proliferate some more and also to become a memory cell. So now memory cells are a hallmark of the adaptive immune system. They're the basis for vaccine development. That is that once a T cell or a B cell gets exposed to an antigen, maybe I better put this on the view, so that it generates cells that have the same specificity. And they're called memory cells because they're long-lived. They're in the body for a long time. Now, I should get off of this because I want to show you a little movie that tells you the important role of the dendritic cells in this process. So let me go get that. Wrong. I had already queued it up and now I don't know where it is. Give me a minute here so I can find it. As you can see, my screen is way too busy. Where's the movie? I should have closed all these other screens. So, oh, here we are. Okay. So I'm going to show you this movie. It's very short. And I hope, see, this is my problem. It won't.
Just try clicking on the screen and then.
Here we are, right here.
Dendritic cells, the key antigen-presenting cells of the immune system, are generated from progenitors in the bone marrow that migrate into peripheral tissues through the bloodstream. There, the immature dendritic cells lie in wait for pathogens entering the body, through sites of injury, for example. Dendritic cells express various pattern recognition receptors that can recognize common features of many bacterial and fungal pathogens. Through these receptors, they're able to bind to and phagocytose pathogens. When these receptors bind pathogens, they activate the dendritic cells, which then start to mature. In this process, they migrate from the tissues and change their behavior to stop phagocytosis and to start expressing immune stimulatory molecules. The activated dendritic cells migrate from the tissues into lymphatic vessels, where lymphatic fluid drains through lymph nodes, carrying the dendritic cells with it. T cells, migrating through the lymph nodes, inspect the dendritic cells for the presence of antigen. T cells that fail to recognize antigen on one dendritic cell carry on to inspect others, and eventually return again to the circulation. T cells that do recognize their specific antigen become activated, and both proliferate and differentiate into effector cells.
Okay. So now I want to get out of my full screen. I'm a real idiot about some of these things, I'm sorry to say.
Hit your escape button. Should take you out of full screen.
Okay, great. Okay, so that tells you the importance of the dendritic cells. So then the next step that happens, why did it do that to me? Oh yes, so one thing I wanted to say is that for RNA viruses, such as coronaviruses, the receptors for the virus that activate the dendritic cells actually aren't on the cell surface. They are molecules that exist in endosomes and these intracellular vesicles in the cell. And there's one that TLR3 that recognizes double-stranded RNA, and TLR7, which recognizes single-stranded RNA. Coronaviruses have a single-stranded RNA genome. If you think about this, is really clever because the virus has to be internalized in order to replicate. And for coronaviruses, that entry goes into the endosomes because the acidic pH of the endosome is required to open up the virus so that it can replicate its genome. And when it does that, when its genome is exposed, then that genome can be bound by these toll-like receptors. So it's how the immune system has evolved to take advantage of the viral life cycle. So then, while at the same time that these helper T cells are being activated, antigen that comes into the lymph node can also activate B cells. And for these B cells to proliferate and differentiate into either memory cells or plasma cells that secrete antibody, they need to see both the antigen and to get cytokines from the helper T cells. Parenthetically, I want to tell you that this type of cell is called a CD4 T cell, and you might have heard of that term because that's the type of cell that gets infected by HIV. And the fact that HIV infects CD4 positive helper T cells is why AIDS can develop. If you take out this cell, if you kill off this cell, then you basically don't have an immune response because all of the other cells, adaptive cells of the immune system, are dependent on its cytokines. And not only do B cells need cytokines from the helper T cell, but so do the cytotoxic T cells that kill virally infected cells. So at the same time that this antigen comes into the lymph node and stimulates helper T cells and B cells, the dendritic cells also present antigen to type of cell that's called a CD8 positive T cell or cytotoxic T cell. That gives rise to either active cytotoxic T cells that can kill virally infected cells or to memory T cells. So then, the next time you get infected with that pathogen, you have this huge army, expanded populations of memory B cells, memory helper T cells, and memory cytotoxic T cells that can immediately attack the pathogen as it enters the body. And this is the basis of immunization with vaccines. The idea is that instead of going through a first illness, you generate all of these memory cells that are then ready to protect you when you finally do get exposed to the pathogen for the first time. Any questions on that? Okay. So memory is very important because not only are you poised to respond to the pathogen rapidly, you also have a greater amplitude of response. So here is an experiment in which animals were immunized with a particular antigen, antigen A. You get the kinetics of the response are such that you don't see antibodies in the serum until about 10 days. And the amplitude of that response is rather low and it wanes. If you get exposed to that antigen again, although this response waned, there's actually a huge army of memory B cells and T cells. And so then when you get exposed to the antigen again, it only takes 48 to 72 hours to launch a response, and it tends to have a much greater amplitude and it's much longer lasting. I couldn't find a slide that would illustrate what I want to say to you now, which is that in a natural infection, if the antigen isn't cleared right away, then you don't see this dip, right? If you keep on generating antigen, then this is just going to keep going up and up and up. Now, you generate, this is important for vaccine development, you generate two different kinds of antibodies in the primary response and the secondary response. And I'll come back to these later. The one that you generate in the initial response is called IgM. The one that you produce later is chiefly IgG. But at mucosal surfaces, there's a really interesting antibody produced called IgA. So what is the value of IgM and IgA? IgM and IgA are important because they have far more antigen binding sites. So an IgG has only two antigen binding sites, one here and one there. But IgM, because it's pentameric, there's 5 of these immunoglobulin molecules linked to each other, you have 10 antigen binding sites. And the dimeric IgA has 4 because you have dimers of IgA, so you have 1, 2, 3, 4. The other thing that's good about IgMs and IgAs, but especially IgAs, is that they can cross mucosal membranes, and I will come back to that in a minute. So at mucosal epithelia, such as in the nose or the lungs, this is where the pathogen would enter across the mucosal layer. So this is the inside of the body, that's the outside. And this is how pathogens enter. But if you have generated an IgA response, the interesting thing about IgA is that they can cross this epithelial cell barrier by binding to a special receptor. And that means that they can come out to the mucosal surface where they're shed. But now if virus comes in through that mucosal surface, it gets sopped up by the antibodies so that you don't get infected. This is a really important point, so I want to make sure everybody got it. If you didn't, I'm happy to go over it again. Okay. So I'd like to turn to the virus now. Coronaviruses have genomes. This won't mean anything except to molecular biologists like Barry. They have a genome of about 30,000 nucleotides. It's a single-stranded, positively stranded RNA. And here is the virus itself. Its genome is off here on the right. And the virus has these, it's an enveloped virus, so it has a lipid bilayer that is derived from the host membrane. And these red tulip-like structures are the spike protein, which is, I'm gonna have to close this so I can see my own. So Here's the virus, and it binds on the host cell membrane to a protein called ACE2. And it turns out that this needs the cooperativity of an enzyme, a protease, that cleaves the S protein and helps open up the binding site by which it binds to ACE2. So it's not sufficient for a cell to express, a host cell to express ACE2. It also has to have this protease for the virus to infect. Okay. I thought I'd give you a few numbers. In cell culture, coronaviruses, here we use SARS-CoV-1 to determine this. It takes about 10 minutes to enter the cell. It takes about 10 hours to make a population of intracellular viruses, and each cell produces about 1,000 viruses. This was measured for a mouse coronavirus, not for SARS. And if we look at the sites in which SARS-CoV-2 RNA is found in the human body, based on RNA by PCR, there's between 1 million and 10 to the 9th RNA molecules per swab in the nasopharynx. So if you go get tested for the COVID test, this is what they're measuring. In the throat, there's about 2 orders of magnitude to 1 orders of magnitude lower. It's found in the stool as well, and then in sputum. And the sputum actually has the highest amount of virus. This is probably why some nurses, if they don't have proper PPE, are so readily infected, because of the virus coming out in the sputum. And then here are the characteristics of an infection in a single patient. So there's a latency period of about 3 days before you see symptoms. And the patient, usually diagnosis can happen after about 5 days. That's when the patient feels sick enough to go in. It takes a total of about 2 weeks to recover from SARS-CoV-2 infection if you have a mild case of it. If you have a severe case, it can take 6 weeks. Here it says about, but I know of patients who have taken literally months to recover. They may not have the fever and some of those other overt symptoms, but they're still not well. You may have noticed this case fatality rate is 1 to 15%, an infection fatality rate. What this means is the number of people who died relative to the number of people who came into the hospital sick. And then this infection fatality rate is the number of people who died relative to all of the people who got infected. This number is lower because, of course, we have a lot of people who have very mild symptoms and don't go into the hospital.
Martha, I have a question that people ask me is, how many viruses does it take to get an infection? I mean, if you got one virus, you're unlikely, right? But
oh yeah, absolutely, you're unlikely to. I don't think it's known how many you need to be exposed to. That question maybe could be answered in the studies on rhesus macaques, where you can willfully infect them with different amounts of virus. I'm not sure, actually, but I would think you'd have to be exposed to thousands or even millions of viruses. But I'm not sure. I don't think we know that. One number I forgot to mention to you is this. This is the number of people, R naught measures the number of people who can get infected from a single person. And the average is 2 to 4, but you know, some of these studies that have been done on cases where somebody had a wedding, maybe you all heard about that wedding in Maine, in which there were 75 guests, and in the end, 200 people, none of whom were at the wedding, ended up hospitalized, and there was some people, I think a dozen or so people who died. So it can vary quite a bit, but the average is 2 to 4. This is far lower than measles virus. Measles virus is one of the most infectious viruses known to man, and in that case, one person can infect literally hundreds of people. That's why there were those outbreaks of measles at places like Disneyland, because somebody who has measles can infect basically practically everybody who's there. So this is a blessing. This number is relatively low. Antibodies appear in the blood after 10 to 20 days. We don't know how long you maintain an antibody response to SARS-CoV-2, nor do we know if that antibody response is protective. For SARS-CoV-1, it seems that the antibody level in the bloodstream is about 2 to 3 years and is protective. But we've been hearing about people who have SARS-CoV-2 getting reinfected. And if we have time at the end, I'll come back to some of the nuances of that observation. Here's some information on the viral environmental stability. In aerosols, the half-life is about an hour. On surfaces, it's said to be 1 to 10 hours, but I want to point out something. Some of these studies were on quantifying infectious virus, but some of them weren't. They were just measuring whether they could find RNA there. And just because there's RNA there doesn't mean it's infectious. And in fact, the most recent studies have suggested that although you can find infectious virus as long as 10 hours afterwards on surfaces, that's RNA, it's not infectious virus. So you don't need to worry about when you go get your groceries, not touching them for 10 hours before you unpack them or anything like that. Okay. Are there any questions before I go on to my next section? If not, I'd like to talk about how the virus comes in. It comes in through the nasal passages, and it turns out that here we have in our throat, we have organized lymphoid tissue, our tonsils and our adenoids. And that mucosal tissue is the first way that dendritic cells can encounter the virus that enters the body. So underneath the mucosal epithelial cells here, we have organized lymphatic tissue. And I want to show you this little sort of cartoon cross-section of mucosal epithelium. Here, these gray cells are the epithelial cells. Then there's these very fascinating specialized epithelial cells that are called M cells that have these huge pockets that are important for uptake of pathogens. And then this is my favorite part. The dendritic cells at mucosal surfaces put out these long arms that reach out to grab antigen. So they can sample antigen. And then if they get activated, they retract this process and they migrate to lymph nodes just the way the little movie showed you. But, oh, so there's one thing, there's a problem with this.
May I ask a very simple, would you please define mucosal?
Mm-hmm.
So that's a great question, Rusty. Mucosal epithelia are the epithelial surfaces in the nasal passages, the throat, the lungs, the vagina, the intestine. These are all tissues that have contact with the outside world, and the mucosa that are produced by the epithelial cells, which the mucosa surface isn't really shown here, actually traps pathogens. The idea is to keep them from entering. Also, those IgA antibodies that I told you about, they reside in that thick mucosal, that mucus that's, oops, sorry, didn't mean to do that. The mucus that's produced by these cells and helps keep those IgA molecules there so that they can capture viruses and bacteria that come in through the surface. Did that answer your question?
Yes, thank you.
Okay. So, so when coronavirus comes in here, it can be taken up by these M cells, it can be taken up by these dendritic cells, and it can launch the first phase of the immune response, the innate immune response. And I just want to show you here, excuse me, the kinetics of an immune response after virus infection. There's kinds of cytokines that are called interferon alpha, interferon beta, TNF alpha, and IL-12. And these are all produced very quickly. As you can see, you start to see their production within 1 to 2 days, and they peak at about 2 days, and then that response wanes. But these cytokines actually activate other cells of the immune system, including an interesting innate immune cell called a natural killer cell. And natural killer cells get activated, that's the blue peak here, and they peak at about 3 to 4 days, and then the response wanes. Finally, because of this activation, so I'm just going to go back a few slides here just to remind you. Oh, maybe I have to go back lots of slides. Sorry, I should have put another one in there. Well, here, when the dendritic cells bring in the antigen and they migrate to the lymphoid tissues, this proliferation of the B cells, helper T cells, and cytotoxic T cells all takes about 7 days. And so that's why you don't see, the antibodies against the virus peaking until about 7 to 10 days, and that's this red peak. So in an ideal situation, you need these interferons, the natural killer cells, and the cells of the adaptive immune system to cause your virus levels to go down. So this yellow peak is the virus titer. You can see that it's plateauing after the natural killer cell response has waned, but to completely eliminate it, you need the adaptive immune response, and that brings it down completely. And for the average virus, that takes 10 days to 2 weeks. So if you get the flu, you're not really well for at least 10 days. Now, here's the problem. I told you that dendritic cells pick up the virus. So here the virus is in red. And so in the nasal mucosa, the virus can be picked up by these dendritic cells, which are green. And you can see that the dendritic cells pick up the virus, and then they go to the tonsils and the other lymphoid tissue. But if the virus isn't eliminated, if it can replicate there, then basically the dendritic cells are unwitting Trojan horses that bring the viruses deeper into the body. So the dendritic cells both initiate an adaptive immune response, but they also kind of are traitors because they bring the virus deeper into the airways. And there is a major problem, of course, right? And this is one reason it would be very good to develop vaccines, because if the antibodies of the vaccine can bind to the virus out here on the outside of the mucosal membrane, then the virus will never get picked up by the dendritic cells and won't go in and carry it deep into the tissues. Now, several slides ago, I told you that ACE2 is the receptor for SARS-CoV-2. And one thing that was puzzling initially after the first cases of COVID-19 developed was, it was surprising that so many people developed diarrhea and other gastrointestinal problems. And we now understand why, because ACE2 is expressed at high levels in the duodenum, in the small intestine, at low levels in the colon, but at high levels in the kidney, high in the gallbladder. Interestingly, in the heart, ACE2 levels are high only in patients who have hypertrophic cardiomyopathy. So people who have heart conditions are more likely to develop serious COVID-19. And in the lung, it's not expressed, the ACE2 is not expressed in all of the cells, but it is expressed in a cell type that's called alveolar cells. So that also is another reason that the virus can cause such serious pulmonary problems. Are there any questions on that?
Well, I guess I had thought and thinking that the lung was the primary target of this virus. Is that not correct?
Such an interesting question, Barry. The pathology is chiefly in the lung in people who get serious COVID-19. So let's just distinguish here for a moment that SARS-CoV-2 is the virus, And COVID-19 is the syndrome, the pathology that leads somebody to have to be hospitalized. So in people who end up with a mild infection, it never really makes it deep into the lung. But in people who develop serious COVID-19, yes, the infection is chiefly in the lung. There are patients who have reported that they lost the smell. That's because of the infection of the nasal epithelia, that they lost taste, and that they also had gastrointestinal problems, but they never ever developed respiratory problems, which is interesting, right?
Mm-hmm. Mm-hmm.
So now, any other questions?
So I had a question. You said that when you were talking about the dendrite cells that were, or dendritic cells that were bringing the virus to deeper tissue, I presume they're doing that because they think it's going to be somehow destroyed in the deeper tissue, and so obviously that doesn't really happen, right? And why doesn't it happen?
Yeah. So I'd thank you, Kitty, for that question, because I think that I didn't state clearly what I meant. I was kind of sloppy with my language. So the dendritic cells take the virus into the tonsils and into the adenoids and the other lymphoid tissue that's in your throat. But the problem is that if the virus can replicate there, then that virus itself, just by coughing and all of the other distress that you're experiencing while you're infected, can cause, the virus can drain into, not necessarily being carried deep into the lung by the dendritic cells, but can seep down into the lungs. So you can, I'm not aware, I was wrong in the way I stated it. I'm not aware of dendritic cells that actually travel into the lung. The dendritic cells travel to the tonsils and the adenoids and the other lymphoid tissue that is associated with the lung, but the virus now is, it's escaped. It's on a highway into the lungs just because of all of the fluid flow from the nasal passages and the
But that must work for some other types of antigens, right?
Yes.
Okay.
So people can end up with pneumonia after they get influenza for the same reason, that the virus can be carried further down.
I have a question.
Yes.
That many of us seniors no longer have tonsils and adenoids, but most kids do. What difference is that making?
That is such an interesting question. I know that I don't have my tonsils. According to the ENT doctor that I saw, I do still have my adenoids. My mother, she had 10 kids, as Barry mentioned, and she decided that her kids were getting too many sore throats. So she just basically took us all to the doctor and said, take out all of their tonsils. And he abided by her wishes. And we didn't get so many sore throats anymore, because a sore throat is literally due to, remember I told you that when the T cells encounter the antigen, they proliferate? You get a sore throat when you get a cold or the flu. Literally, it's because of the pressure of all of those replicating cells making the lymph nodes larger and then putting pressure on neurons, sensory neurons. So the ENT doctor that I saw, when I told him I'd had a tonsillectomy as a child, he said, well, open wide and let me look. And he said, well, fortunately, the doctor took out all your tonsils, but he didn't take out the adenoids, and so you're fine. So I can't answer your question directly, but I'm wondering how common it is. They actually don't take them all out. They leave something behind. But I suppose if all of them had been removed, you'd be at greater risk for something like SARS-CoV-2. Yeah. Scary thought, isn't it?
I have another basic immunology question. How do the dendritic cells know what's a foreign protein and what's not?
So there's these special receptors that are called toll-like receptors. And Barry, you might be interested in knowing that. Did you ever know Charlie Janeway at Yale?
Who was it? Who?
Charlie Janeway.
Oh, no, didn't know.
Okay, so Charlie Janeway was a Yale immunologist, and he basically discovered toll-like receptors in humans and in mice. And there's a whole array of toll-like receptors that have specificity for molecular entities that are sort of global patterns, like general patterns. Bacterial lipopolysaccharide is one. These viral RNAs are another that don't exist on human cells. And so that their toll-like receptors get activated by these pathogen-produced molecules and not by our own. These molecules don't exist in our own tissues. So that's how they get activated. Like those TLR3s and TLR7s that I mentioned earlier. Does that make sense?
Yes.
Okay. All right, so I wanted to talk about cytokines. Cytokines are a family of relatively small molecules that are produced by cells of the immune system and some other cells in the body, and they work in there's three different classes, and they're categorized by how they function. So autocrine cytokines are cytokines such as the IL-2 that I mentioned before, in which the cell that produces it also has a receptor for it, and so it can stimulate itself to do something. IL-2 stimulates T cells to proliferate. But IL-2 is interesting because not only does it work in an autocrine fashion, it works in a so-called paracrine fashion, in which it can bind to receptors that are nearby. So for example, a T cell that's producing IL-2 can stimulate the proliferation of cells that are in its neighborhood. Now, cytokines, in a perfect world, this is how cytokines work. One way to think about it is, let's say that you're sitting in a lecture hall and you want to, Rusty and Barry have gone to the theater, and Barry remembers something that he wants to tell Rusty before he forgets, and so he turns to her and he whispers in her ear. The information that he's telling Rusty, only Rusty hears. None of the people around Rusty and Barry hear that. That's kind of the way cytokines work. The immune system is evolved so that a T cell is sharing its cytokines only with the cells that it wants to activate. And in that manner, immune responses are regulated and contained. But there are some cytokines that are called chemokines that I don't have time to talk about today, that work in a so-called endocrine fashion. They're secreted and they go out into the bloodstream, and then they can have activity on a distal cell. Now, one problem, the whole basis of cytokine storms is that if too many T cells get activated, they produce so much cytokine that it doesn't all get sopped up by the nearby cells, and it can get into the bloodstream and then activate all kinds of other cells. So going back to my theater metaphor, if Barry said to Rusty, "I think I smell smoke," but he just whispered it, nobody's going to get excited. But if he yelled it out, "I smell smoke," you might have everybody in the theater jump up and run away because they're worried that there's going to be a virus or something, a fire. So cytokine storms are basically an overly exuberant production of cytokines so that you activate all kinds of cells in a non-regulated manner. Does that make sense? Are there any questions on that?
Well, then what's the consequence? I mean, what do these activated cells do that makes it a bad outcome?
It depends on the cytokine, and I'll come to that probably in the next slide or two.
Okay.
So first of all, before I get to that, I just want to point out the kinetics and the intensity of I can't read this because it says, I can't read the title of my slide, but anyway, the kinetics and intensity of the immune response and of the cytokine production is decisive in the outcome of a SARS-CoV-2 infection. So under normal circumstances, Donald Trump, for example, who got the best medical care in the world. If you get infected, you get your, so here's your type 1 interferon response. Those were those cells that the cytokines get produced at the very beginning. You have a peak of it, but then in the meantime, you're activating lymphocytes and they are proliferating and getting rid of the virus. And your inflammatory cytokines, which I'll come back and talk about in a minute, have their peak early in the infection, and then their concentration wanes. So that in a normal situation, you have mild or moderate disease, you produce your type 1 interferons quickly, you also amplify the production of these lymphocytes, and so the disease wanes, in keeping with also your cytokines waning. Now, in people who get severe disease, and there seems to be there's several factors that affect whether you get severe disease or not. One is the role of the genetic dice. It turns out that your ability to produce type 1 interferons and the genes that are downstream of it, for them to be activated quickly, that there are some mutations in the regulation of this. And so some people do not produce them rapidly enough. But there's also the problem of pre-existing conditions. If you are obese, or if you have certain autoimmune disorders, or if you've been smoking, these cause you to have a state of already elevated inflammation. You are actually already have inflammatory cytokines coursing through your body. Obesity is a particularly bad one. We now understand that inflammation in obese people is at a high level in the absence of infection. So what seems to happen in severe disease is, notice this green peak here, that's your type 1 response. Instead of occurring within a day or two of an infection, it's delayed. So this peak is shifted over to the right. And because it's delayed, the virus can replicate with wild abandon. But then these inflammatory cytokines also are produced in a dysregulated manner. And we're just beginning to understand why that's the case. But I just want to mention that there are already some antivirals that have been used to treat autoimmune disease already for more than 20 years that are available to treat patients. And I'm going to talk about a few of those. I think it's on the next slide or two. So this slide shows the most important pro-inflammatory cytokines: interferon gamma, TNF alpha, IL-6, IL-1 beta. This one we're not going to talk about. And these all are very important in getting rid of viruses. Interferon gamma activates natural killer cells and also macrophages to kill virally infected cells. TNF alpha also potentiates the ability to kill virally infected cell. IL-6 and IL-1 beta stimulate adaptive T cells to kill virus. So under normal circumstances, you want those guys. But unfortunately, if they're produced in an exuberant manner, this sustains an inflammatory milieu. IL-6 in particular stimulates the production of more TNF alpha, which affects blood coagulation and all manner of other physiological effects. If it's present at too high levels, it promotes edema. It's just a bad boy if it's produced in too high an amount. And interferon gamma can produce pulmonary inflammation and pathology if produced in the lungs at too high an amount. So basically, if you remember the story of Goldilocks and the three bears, and Goldilocks wanting to eat the porridge that was just right, the immune system is kind of that way. You need these cytokines to be produced just in the right amounts and in the right locations so that you can fight the viral response. The augment the viral response. If they're produced at the right time, then you end up with this happy situation where the virus is cleared. If they're produced at an exuberant response, then everything goes south very quickly in the patient. And this is what we observe chiefly in people who have been smoking, who are obese and have other pro-inflammatory pre-existing conditions. This can also happen in the elderly, and we don't know why. We don't know if it's because the type 1 response just isn't as good as it would be normally. That is not well studied.
So a question on that is, over the years, interferon has often been thought of as maybe a great drug, but it's never worked out. So
Yeah. Oh, Barry, I'm so glad you asked that question, because I was going to talk about that and then thought I might not have time. But it's actually not the interferon gamma that's been used, it's interferon alpha beta. So Barry, do you remember Wally Gilbert?
oh yes, yes.
So Wally Gilbert was a Harvard professor, and he started a little company called Biogen. And back when you and I were at Yale, I remember the McNeil-Lehrer NewsHour on NPR interviewed him because Biogen had made recombinant interferon. And he very smugly, Wally knew less immunology than a potted plant does. So was really speaking just out of arrogance, not out of knowledge, but he said, this is going to cure cancer, it's going to treat all these diseases, everything's going to be fine, and doctors are going to be out of a job. Well, it turns out, remember this slide? Under the best of circumstances, the cytokines act only on the cells that are nearby, right? So IL-2, for example, acts on the cell that's producing it and on nearby cells. It turns out that when they first were giving the interferons, the Biogen produced interferons, they ended up with a terrible, life-threatening situation that was essentially a cytokine storm. The injection of the interferon was such that it was working in a paracrine fashion, and it was having effects, sorry, I didn't mean to do that, it was having effects on all kinds of cells at distal sites. So cytokines are used to treat diseases. For example, interferon alpha beta is used to treat people who have multiple sclerosis. But it's got serious side effects because of this particular problem. Basically, we haven't been able to really recapitulate what the cells of our immune system do by themselves quite nicely. We haven't figured out a way of producing cytokines such as the interferons in such a way that they'll act in this regulated manner illustrated in this slide. I don't know how you would do that, frankly. Little time release capsules or something, I just don't know.
May I ask a question?
Yes, please.
I'm a little confused. Tell me the difference between cytokines and interferon.
Oh, interferon is a class of cytokines.
Okay.
I'm sorry. So there's a huge family of cytokines. The number is, I don't know, there's at least 30 or 40 of them probably. The ones that we worry about with regard to, interferon gamma is a cytokine, TNF alpha is a cytokine, IL-6 is a cytokine, IL-1 beta is a cytokine. Cytokines are this family of molecules that have these profound biological effects. They evolved so that cells of the immune system could do their jobs, right? But so, for example, one of my favorites is interferon gamma. It's produced by CD8 positive T cells and also CD4 positive T cells, and it can activate macrophages and natural killer cells to kill virally infected cells. So in terms of virus infections, interferon gamma is our best friend, but not if it's produced in an overabundance. Did that answer your question? Yeah.
Sort of. So interferons are really the workers. Is that what you're saying?
Okay, so I don't want to sound like an interferon gamma chauvinist. All of these guys are workers. All of these guys are hard workers.
Right, sure.
Martha, I think of them basically as signals, right? They're a signal from one cell to another to turn it on or turn it off. Is that right?
Yes, exactly. So for example, if we come back to this slide, cells will have cytokine receptors on their surfaces. So that means the cytokine that's produced won't have an effect on a cell unless it has a receptor for it, right? Sure. And when the cytokine binds to its receptor, that will cause an array of events. We call that signal transduction through the cytoplasm of the cell, sending instructions into the nucleus to now express specific gene types. Those genes might cause these cells to proliferate or cause them to become a particular cell type that then produces something. So if we go all the way back to, this is such an important point that I really want to. So for example, the cytokines that the memory helper T cell produces stimulate the memory T cells and the memory B cells to now proliferate and either become plasma cells or active cytotoxic T cells. And one thing that's interesting is there's a whole array of different cytokines, and some of the cytokines stimulate cells to do things, but some of them stop things from happening. So the cytokines are both the accelerator pedal of the car and also the brake pedal. Depending on the circumstances, they can either jazz up the immune response or they can dampen it. Did I confuse you or did that help?
Okay, so that's cytokines, and then the interferons
are a type of cytokine.
Okay, thank you.
So now, I think I got rid of all of my.
Okay.
I think I got rid of all of my slides with macrophages on them, which is too bad. But yeah, I got rid of them. But so, for example, these molecules here, interferon alpha, interferon beta, TNF alpha, and IL-12, these are all cytokines that are very important for the activation of natural killer cells and also for the activation of T cells and B cells. They all have their own targets. One thing that's interesting is that, so let me see. One way that the immune system regulates cytokine responses is it can make the cell shed its receptors. It can make soluble cytokine receptors that go into the tissue spaces and bind up that cytokine. So regulation of cytokine production and cytokine sensing is tight under normal circumstances. It's very tightly regulated. But when you end up with an overly exuberant production of cytokines, then all of those regulatory mechanisms can't keep up. I mean, one way to think about it is if you just have a hose on that's shooting water, or you have a pipe break and now you have, just a flood of water coming. You just don't have enough buckets and mops to clean up the flood of water the way you would if you just had a small leak. Did that help or not?
Well, maybe related to that, is this where the dexamethasone comes in?
Yes. Thank you for asking that. So corticosteroids really put the brakes on the immune system. Corticosteroids inhibit the activity of T cells and of macrophages. So if you have an overly exuberant immune response, treating with drugs like corticosteroids like dexamethasone help lower that inflammatory response. So corticosteroids such as dexamethasone are great anti-inflammatory cytokines. Because they inhibit T cells and macrophages sort of globally, you really don't want to give them until you have evidence that the inflammation is unregulated, because you don't want to prevent this normal immune response from happening. Mm-hmm. But they are a great way to regulate things. Indeed. you know, you can't take them forever because they have other systemic effects on the liver and other organs. They also make you high as a kite. I can tell you that because I had to take them when I was undergoing chemotherapy for breast cancer. And so if you might have all noticed that Donald Trump was just so excited and happy when he emerged from the hospital. He was on a dexamethasone high, I'm sure of it. It makes you feel like Superman. But that's not immunology.
I have a question about vitamins D, C, and zinc. Do those help, and how do they work?
Okay, zinc interferes with the activity of the viral RNA polymerase that replicates the genome. So if you take zinc early enough, it can help limit an infection for many viruses. Don't know if it works on coronavirus, but for many, but the trick about zinc is you really have to take it early. If you already have the sore throat, if you already have the symptoms, it's not going to work all that well. But if you take it early, it used to be that every year when I'd go home for Christmas, I'd hug my little nephews and nieces, and I was guaranteed to get a cold. And so I used to take zinc lozenges with me, and I would suck on them and just sort of try to prevent myself from ending up with a cold. Vitamin D regulates the immune system. People have been studying vitamin D for at least 25 years to modulate immune responses and infection. The problem is that the animal models suggest that the amount that you have to take is just so huge that it has other problems. So yes, in cell culture, you can see all of these wonderful effects of vitamin D, but if you extrapolate to the amount that a human would have to take, it's just too huge. So we don't know how it normally regulates or helps immune responses at physiological levels. I like to think of it in the same way that we think of this, that maybe vitamin D at specific sites can have very beneficial effects, but if you're taking huge amounts of it and it's going into the bloodstream, it won't necessarily have the same effect. Vitamin C is another one. We know that vitamin C, it's good antioxidant, and it does seem to stimulate immune responses, but again, you seem to need to take it at huge amounts, although unlike vitamin D, taking huge amounts of vitamin C doesn't seem to hurt you.
Can I ask about monoclonal antibodies?
Yes. What would you like to know? Do you want to know what they are?
Well, I have a vague sense of what they are, but I remember reading they also had to be administered early to have the effect.
No, that's not true. I mean, it probably depends on the condition, but that's not true. Monoclonal antibodies have been used therapeutically to treat a variety of diseases. In fact, here we are. Almost all of see these syringes here that are pointing towards gamma interferon, TNF alpha, IL-6, IL-1 beta. These are all antibodies that are specific for these cytokines. And they're monoclonal antibodies that were generated in the lab that when administered can bind up the interferon gamma or the TNF alpha or the IL-6 or the IL-1 beta and sop it up so that it doesn't have these deleterious effects. And in fact, antibodies against TNF alpha have been on the market, I want to say about 15 years or so, to treat certain autoimmune conditions such as Crohn's disease and ankylosing spondylitis and rheumatoid arthritis. Did that help?
Yeah, thank you.
Okay. So the pace of SARS-CoV-2 vaccine development has absolutely been breathtaking. It's not because the normal process was inordinately slow. It's more because we've gotten rid of a lot of the, in my opinion, usually necessary guardrails to make sure that things are done properly. I'm particularly concerned about the fact that for this particular round of vaccines, the Moderna one and the Pfizer one, we don't know their long-term effects. We don't know when you get immunized, is that going to give you immunity for months or is it going to be years? So the traditional vaccine development can take up to 15 years because the animal studies usually take 2 to 4 years. The clinical trials are usually 5 to 7 years. The regulatory review by various regulatory agencies can take a year or two. Then you have to scale up for long-term production or large-scale production. In the case of the SARS-CoV-2 vaccine development, they've reduced this to weeks and months. They're doing the phase 1, 2, and 3 trials in overlapping phases instead of sequentially. And the one that worries me the most is that, okay, so let's just talk about the vaccines we know about. If you go get a yellow fever vaccine, the immunity that you get from that vaccine is so good that depending on which epidemiologist you talk to, you need a new vaccine in 12 years or you don't need one for the rest of your life. It's a very, very long-lasting vaccine. The memory from the yellow fever vaccine is very robust and very long-lasting. Now let's turn to our good old friend influenza vaccine. The immunity from the flu vaccine wanes in 4 to 6 months. 4 to 6 months. If it turns out that the SARS-CoV-2 vaccines wane in 4 to 6 months, we need to know that, right? We need to know that so that people can be re-immunized. But the problem is we don't have time to find that out because we're trying to roll these vaccines out as quickly as possible. And I read an article in which a representative from Moderna was talking, and he said that he reckoned it would take them at least 2 years to get really good clinical data on how long their RNA vaccine will last. So when these vaccines are readily available for the general public, you definitely want to keep using your masks. You want to still social distance and be sensible because we don't really know how long the vaccine's going to last. That's really a little bit worrisome. So this slide shows the types of vaccines that are being developed, and I'm not going to have time to talk about the different types. The more traditional methods that have been used for decades are inactivated virus vaccines and also protein epitope-based vaccines. These have been around for about, I want to say, 30 years. Viral vector vaccines, especially the adenovirus vectors, have been around for cancer, for the development of cancer vaccines for probably about 20 years. They have a very troubled history. But I think we've learned from the mistakes that happened early on in the development of these vaccines. And the more recent ones are the DNA and RNA vaccines. DNA and RNA vaccines have been tested in animal models for at least 20 years. But up until now, especially for cancer, but up until now, they've really not been that great. They just haven't. So it's quite interesting to me that's what's going to be rolled out here in the United States. So this shows the SARS-CoV-2 vaccines that have been under development around the world in the past 10 months. Here we have the preclinical candidates. These were tested in animal models, and you can see the majority of them were these protein subunit vaccines, followed by non-replicating viral vectors and by RNA. And then these others, the replicating viral vector and the DNA vaccines, inactivated vaccines, and live attenuated represent a smaller number. So there's a total of 62 preclinical candidates. Now, of the clinical candidates that we have being worked on right now, there's 15. And you can see that the non-replicating viral vector has gained on the protein subunit vaccines in terms of how many. There's 4 of these that are in clinical trials right now, 4 of these, 3 inactivated ones. the 2 RNA ones, which are the ones that we're going to talk about today, and 2 DNA ones. This shows, is color-coded basically what was on the previous slide, just showing you the various stages of preclinical testing. You can see that some of them drop out. The recombinant spike protein vaccines seem to be diminishing at the expense of some of these others, and then shows you which ones are in phase 1, phase 1, 2, and so on. These 2 here, these green ones, so green is the replication incompetent vector vaccines. Both of these received the first licensing. One of them was developed in China for the Chinese military, and the other one was developed in Russia, and that one didn't go through a phase 3 clinical trial. It went straight from phase 2 into human use. Now, one thing I want to talk about here briefly is something that is kind of getting ignored when we talk about the Moderna and Pfizer vaccines. And that is, remember that the infection by the virus is through the nasal passages. It enters through mucosal surfaces. And in an ideal world, we really want to get immunity, production of IgA antibodies in these nasal passages. But when you do an intramuscular or intradermal vaccination, you mainly get immunity further down in the lungs. Whereas if you do a natural infection or you intranasally immunize, you will get immunity, IgA immunity in the nasopharynx region and IgG antibodies in the lungs. And this figure here just reminds you why we want the IgA antibodies produced, because they will cross the mucosal epithelial cells and bind the virus as it's coming in. And I don't know why, I'm not sure why it is exactly, but the RNA vaccines have to be injected. We don't have a way of making them an aerosol that can be inhaled. So what does that mean? I don't know. I mean, I suppose it'll be good enough to have antibodies down here, but I think all of us would agree that if we could stop the virus up here in the nasal passages, we would all breathe easier, and pun intended. One last thing I want to mention before I move on to some of the data of the viruses is that RNAs, as Barry probably knows, are very unstable because there's ribonucleases in body fluids that can degrade them. And so a key development that was not done with regard to these particular vaccines, it was actually done quite some time ago for other vaccines, was to incorporate what are called nucleoside modifications, unnatural nucleosides that make these RNAs resistant to degradation by cellular enzymes. And one thing that we don't know is if those toll-like receptors that I mentioned to you before that sense the RNAs when the virus gets taken up, we don't know if those can sense the modified RNA that's used in the vaccines. This review article that I found indicated that it is not, but I actually haven't seen any scientific data that confirms that it's not. In a perfect world, the nucleoside modified RNA that's used in the vaccine would be detected by these toll-like receptors, so you can get robust activation of the dendritic cells. Another thing I want to mention, and I'm sorry I couldn't find a high quality image for this, is that these RNA vaccines are incorporated into synthetic nanoparticles that have cholesterol and other lipids on them. This is very important because then it means that this can fuse with the membranes of the dendritic cells or even be taken up into their phagocytes, making them, that's actually important because especially if the nucleoside modified RNA can't actually activate the cells, well, it could be that just the fusion of this nanoparticle to the membrane of that cell will activate it. I had to return to Star Wars, so I put my little stormtroopers here in place of the spike protein. So the spike protein is the SARS-CoV-2 protein that binds to ACE2 on host cells, and it normally exists on the surface as a trimer. So here we have the data for the Moderna vaccine and for the Pfizer vaccine. They're both mRNA based. However, the Moderna vaccine is it's just the messenger RNA that encodes that spike protein, whereas the Pfizer vaccine is very clever. It expresses, it is an RNA that causes a trimer version of just the receptor binding domain. So just the portion that will bind to ACE2 on host cells. Both of them have to be administered in comparable doses. The first time you get immunized, the antibody response is pathetically low, but after a secondary booster immunization, at the higher amounts, you do get, not great, actually that's pretty wimpy, but you do get an antibody response. And very importantly, for the Moderna vaccine, they've demonstrated a good CD4 positive T cell response. Remember, these are your helper cells, so you want a good response of those. For some reason, that wasn't tested yet in the Pfizer vaccine. And I just want to remind you that the spike protein mediates the receptor binding and entry into the virus, and that it causes fusion of the host cell membrane to the viral envelope. And it transitions from a pre-fusion to a post-fusion conformation. And the RBD, the receptor binding domain in the Pfizer vaccine is stabilized in such a way that it'll remain in this pre-fusion state and will bind. That's important so that it can bind to the portion of the S protein that would fuse to the host cell ACE2. Is that clear? Did I say that in a clear way or not? Nobody has any questions?
Could you just repeat, could just say it again?
Yeah, so let's see. I wish I had, I don't think I incorporated, well, yeah, darn it. So I'm gonna go all the way back to the beginning because it'll just be faster to say it that way than to try to just talk my way through it for you. So see these tulips? See those?
Mm-hmm.
So these are the three, this is the trimer of the S protein. And the heads of the tulips here, right here, that makes up the so-called receptor binding domain, or RBD. And it has to remain in the pre-fusion state is how it binds to ACE2 on the surface of the cell. So the spike protein, you can imagine that if you made this vaccine and you introduced it, remember it's not on the virus, it's just in this lipid nanoparticle.
Right.
If that protein could open and close, if its RBD could flop around and open and close, then it might not be such a good immunogen for protecting cells from being infected. If you make it stabilized so that it remains in this pre-fusion state, then you're making antibodies against this very shape that would be the shape that would bind to the receptor. So you would basically be preventing infection. Does that make sense?
Mm-hmm.
Yeah. Yeah. Thank you. Thank you.
Yeah. So it's a very clever strategy, and it's what distinguishes the Moderna vaccine from the, the Pfizer vaccine from the Moderna vaccine. Even so, you know, I'm glad that they both have demonstrated 90 to 95% efficacy. These antibody titers are, to use my sister-in-law's expression, not to die for. I mean, you just wouldn't crack open a bottle of champagne if you were a basic immunologist. I guess if you're the company, you do crack open the bottle of champagne, but that's a little low. I would prefer a more, so what that means, I want to explain what this means. So this is called the titer. This tells you how much you can dilute the antibody from somebody's bloodstream. So you've immunized a person with this, and now you take out their blood and you and you take that serum from the blood and you dilute it and see if it can still bind to the virus and prevent infection of cells. And a good titer would be like 1 to 10,000 or something like that. But here they're getting titers of 1 to 654, titers of 1 to 437. It's a C student. It's not an A+ student, you know. Maybe C student is all we need, but it's a C student anyway. So now this busy table, I meant to break it down and then I forgot to. I'm gonna have you focus just on this line. These are the data for the Moderna vaccine. I just want you to know that all of their preclinical tests were done in rhesus macaques. And in these macaques, they were able to look at various dose ranges. And it's interesting that in the macaques, they got a much better response. They got a 1 to 3,481 titer for their antibody. That's tenfold better than they got in humans. I don't know why. I would love to be able to ask them why that's the case. Interestingly and disappointingly, in the rhesus macaques, they did not see protection in the upper respiratory tract. URT stands for upper respiratory tract, LRT stands for lower respiratory tract. So they didn't, in other words, they weren't finding antibodies up there. So that's not surprising, because remember, I told you that if you do an intramuscular injection, you have the antibodies mainly down here in the lung, and they're IgG antibodies. You need to do an intranasal vaccination to have the antibodies in the upper respiratory tract. So these are just clinical data that confirm that that's the case, that they have complete protection at 100 microgram dose on the rhesus macaques in the lower respiratory tract, but they have very little protection in the upper respiratory tract. And for some reason, I can't find these kind of data for the Pfizer vaccine. I even looked at 2 in the morning this morning, hoping that there was a new paper published, but I just couldn't find anything on that. So that's my presentation. And I was wondering what else you'd like me to tell you.
I have a question just from my limited experience trying to compare the two vaccines. When I first heard that they both require 2 doses. Mm-hmm. and then I heard that the Pfizer vaccine has to be held at -70 degrees Fahrenheit.
Actually, even the Moderna vaccine should be held at that low temperature. So these I don't know if it's because of the instability of the, RNA doesn't need to be at -70 degrees C to be stable, but it could be that these cationic lipid cholesterol nanoparticles are unstable at higher temperatures. So I think for them, is it the Moderna vaccine? I'm trying to remember which one. One of them is stable, for months at -70, is stable for up to 5 days at 4 degrees, is stable for less than an hour at room temperature. And then was it Pfizer that you said? Which one did you say is?
Yeah, what I've heard on the radio is Pfizer has to be held at -70 degrees Fahrenheit, which is a cryogenic handling, right. Whereas Moderna can be held at freezer temperature, 30, 20 right. degrees Fahrenheit.
Yeah.
For weeks. So just based on that, I was favoring the Moderna. But I'd like your assessment. If you had to choose between one of the two, which would you choose? And would it be, is there danger in taking them both?
Oh, I seriously doubt there's a danger in taking them both. They both give you a response against the spike protein. I like the fact that the Moderna vaccine, that the company reported the T-cell responses. I wouldn't want to choose now until I know what the T-cell responses are for the Pfizer vaccine. And let me explain why. If you don't get, I'm going to go all the way back to the beginning here. Okay, if you don't get a good memory helper T-cell response, then you're not going to have a good memory B-cell response. And so it could be that the vaccine, well, that'll be like the flu vaccine. In the flu vaccine, we don't get a good helper T cell response, and that's why the immunity wanes in 4 to 6 months. So what would make me choose which vaccine I wanted was I would want to know, do they both give you a good T cell response? And do they both give you memory? And we don't have the data on that for Pfizer yet. It's kind of interesting that when I've looked up the papers, you know, it's kind of laborious. You have to find the authors. They don't actually, they're not researchers who are at the companies. They're researchers who collaborate with the companies. And so you really have to read you have to dig deep in the weeds of the methods to figure out which vaccine they're talking about. And so I might have missed the Pfizer data, but the Moderna data were easier to find.
So the way I interpret your answer, and thank you very much for the answer, is that, excuse me, I would trust Moderna more than I would Pfizer.
At this moment in time. But that could change if in a month or in a week, Pfizer comes out with its data.
Yeah.
In other words, I guess what I'm saying is that fortunately you and I don't have to make that decision right now, because by the time the vaccine is available for us, hopefully Pfizer will have published these data.
What about safety data?
You know, that's very interesting. We don't know anything about safety in humans. I mean, in the rhesus macaques, it seems to have been safe. But yeah, we need longer, and even for the rhesus macaques, we need longer term data, but you know, on first principles, I think they're probably safe. It's kind of surprising that the side effects at the higher doses, so we need to immunize at the higher doses for both of these. And at the higher doses, the side effects are not life-threatening by any stretch of the imagination, but they're noticeable. I read someplace that they tried even higher doses than these, but they were intolerable. You know, if somebody, how many of you have gotten the shingles vaccine, the newest shingles vaccine?
I did.
So I got, you know, the new shingles vaccine you get in 2 doses. And I got flu-like symptoms from both times. And both times I really didn't feel like leaving my house for about 3 days. I did get out of bed, but you know, I padded around in my jammies and drank hot tea. I didn't do much. And then for me, for those vaccines, that would be tolerable. But if you felt really lousy for 2 weeks, I don't know if people would be very compliant.
Martha, so what would be causing this, you know, not feeling well with the vaccine? What is it triggering? Is it triggering immune response, or?
It is triggering cytokine production.
Oh.
You know, when you get sick, like if you get the flu, the aches and pains, the fever, all of that, that's not caused by the virus. It's caused by your inflammatory response against the virus. TNF alpha promotes fever very, very well. And for those of you who have had the flu or the colds, have you ever noticed that you feel worse at night, and that if you have a fever, your fever's higher at night? That's because we produce our pro-inflammatory cytokines at higher levels at night. We have a diurnal cycle for them, and we produce them at higher levels at night than we do in the day.
Right. So a question on that. So in the typical vaccine in the past, you would be injecting an antigen into a person. But in the mRNA, you're injecting the mRNA that then has to go into a cell, that then has to be translated into a protein, that then has to leave the cell. And so do we have any sense of how long it takes from injection until you actually get antigen circulating in the blood? But the response that they're getting, if they have a side effect, seems to be a same-day response.
No, because remember did I understand you correctly? You mean the side effects?
Yes, yes.
No, because the side effects, okay, so let's think about this. So let's pretend this is a dendritic cell, and the vaccine the nanoparticle that's got the RNA in it fuses with the cell membrane and is taken up. The dendritic cells will migrate to the lymph nodes within, they start leaving the mucosal lymphoid tissue within minutes. They're in the draining lymph nodes in a matter of 20 to 30 minutes, maybe an hour. It doesn't take them long to get there. And then there they can stimulate the production of these pro-inflammatory cytokines, even if they haven't yet started to make the viral protein. It's because the dendritic cells got activated that they can initiate this cascade of cytokine production.
Okay.
But to answer the other part of your question, to the best of my knowledge, it takes several hours for the RNA to be load onto the ribosomes and be translated and to make enough antigen that will then be produced at sufficient amounts that you're going to get a B cell and a T cell response.
Okay. Other questions for Martha?
But I have one. When you talked about the shingles vaccine, some people have a large response to it and some people essentially have no response.
Yeah, so here's the thing that makes me so frustrated about vaccines. So the older you get, the less robust is your adaptive immune response, including a response to vaccines. And it's quite interesting that with regard to the influenza vaccine, you're kind of geriatric by the time you're 35. That is, that when they have measured the immune responses to the influenza vaccine in children, in teenagers, and young adults, and middle-aged adults, and old adults, they begin to see the response, the proportion of people who don't give you a protective immune response jump up in the mid-30s, which is shocking. It's absolutely shocking. And for the last 25 years or so, they've people who care about this have been working on, so remember I told you that, let's go to the beginning. The very first step in the immune response is for the antigen to activate these dendritic cells. There's some basic immunology data as well as a little bit of clinical data that indicates that the culprit of not getting a good immune response to a vaccine has to do with the activation of the dendritic cells. And so they've been working on adjuvants. Adjuvant refers to a mixture of molecules that is produced by a vaccinologist to make a vaccine that will activate these dendritic cells. So it's various molecular entities that are made by bacteria and viruses to goose these dendritic cells. So they've been working to see if they can come up with concoctions that will work on older people. And again, we're not talking about really old. We're talking about people in their 30s with regard to the influenza vaccine. And it's kind of disappointing. I think that their theory about dendritic cells being the culprit must be wrong, because none of their fancy adjuvants have really changed things very much. Now, one thing that's very important about the Pfizer vaccine, I don't remember if it's true for the Moderna vaccine, but for the Pfizer vaccine, wrong slide, for the Pfizer vaccine, they tested people in all different age groups, including people over 65, and claimed that they got a good response in them. And I'm pretty sure Moderna did too. I mean, all companies that are making vaccines are really looking at the high-risk groups, which includes people with pre-existing conditions and people who are older. And so I think we'll have good clinical data for that.
Okay, I'm going to give I'm going to give Rusty the last question.
Okay, Martha, I just have a question about reactions to these vaccinations. I'm one of those who has essentially no discomfort from shingles vaccines or high-dose flu vaccines. Does this mean my immune system is weakening?
No, not at all. Just means you're lucky that you're not producing overly exuberant cytokines. In fact, I would argue, if I can find that slide, I would argue that you're less likely to fall into this group with a severe disease. You'd be more likely to fall into that group.
Okay, that's good.
Well, thank you very much, Martha. This is it's such a fascinating molecular biology topic, how this immune system works.
Well,
And,
I want to thank you, Barry, for inviting me, because I've really enjoyed this, and I hope that I didn't confuse you all and that you learned something from me.
Thank you so much.
Oh, thank you.
I'm going to
Thank you.
stop
Okay.
your screen.